mentioned, I'm a marine biogeochemist and earth historian, and I think about the coevolution of life and environment on our planet, in particular, how cyanobacteria came to oxygenate Earth's atmosphere. And more recently, I've been thinking about how these relationships might play out similarly or perhaps differently on other habitable worlds as well. And so that's going to be the focus of my talk today, is marine biogeochemical cycles on exoplanets. But a fun surprise as I've been exploring these relationships has been that it's actually starting to feed back into my thinking about early Earth and the evolution of our own planet. So towards the end of my talk, I'll highlight a couple of examples of that as well. All right, so before I begin, this is how I begin all of my talks in this new work from home era. I have a 130-pound dog, who you can hear right now, and a grumpy old man cat. And they do not get along particularly well at all, except when they are conspiring to crash my Zoom calls. And they have both done so in increasingly creative ways. And so if either of them make an appearance, my apologies in advance. All right, so as many of you know, I'm sure, our search for life at the moment is effectively synonymous with the search for liquid water. And this effort is really guided by what is known as the habitable zone concept. And the habitable zone is simply the region surrounding a star where it is plausible that a Earth-like planet might have liquid water at its surface. Further out, it's too cold. Closer in, it's too hot. But I think it's important to emphasize that a planet residing within the habitable zone does not necessarily have liquid water at its surface. And if a planet within the habitable zone has liquid water on its surface, that does not mean that that planet is actually a good place to live. The reason being that life requires so much more than just liquid water. Life also requires a source of energy to perform its metabolic activities. It requires a source of carbon to build its biomolecules. And life requires nutrients to provide essential functions within enzymes and other biomolecules as well. And so in theory, any of these ingredients can be limiting for life locally. And in fact, there are environments on Earth where each of these is in short supply. But on a global scale, it is nutrients that limit biological activity here on Earth. And I think that this is going to be broadly true on habitable exoplanets as well. That's in part because the boundaries of the habitable zone as we understand it are really defined by installation and carbon-based feedbacks that act to stabilize global climate and permit liquid water. And so water, energy, and carbon are kind of implicitly accounted for within this habitable zone framework, but nutrients are totally unaccounted for. So when I talk about nutrients, what I am referring to are things like nitrogen and phosphorus. These are the two most important nutrients here on Earth, and that's because they are both important components of molecules like ATP, which is the energy currency of cellular life, and they are both major components of DNA, which encodes genetic information. And so these are both absolutely essential molecules that both require relatively large amounts of nitrogen and a bit of phosphorus. And so nitrogen and phosphorus are the two things that tend to be limiting in this recipe for life. Now, there's a lot of there's a lot of nitrogen in Earth's atmosphere. That nitrogen is triply bonded as dinitrogen, and so it's difficult to utilize, but life has figured out a way to do that. And so on global average, it's actually phosphorus that limits biological activity, not nitrogen. And that's because phosphorus lives in rocks, and it's simply not very soluble. So here on the left, I'm showing a offensively simple cartoon of Earth's phosphorus cycle. And so phosphorus is living in rocks, predominantly on the continents, and that phosphorus is then weathered and transported to the ocean through rivers. And then on geologic timescales, the burial of phosphorus in marine sediments necessarily balances this influx so that the concentration of phosphorus in seawater can achieve a steady state. But before that phosphorus leaves the ocean environment, it can actually be used by life many, many times, perhaps 1,000 times or more before it leaves the surface environment. And so when we're thinking about the phosphorus cycle and its interactions with life, we need to be mindful of both just the total source of phosphorus to the ocean, but then also the extent to which that phosphorus can be utilized repeatedly through recycling. Now on the right, I'm showing a cross-section of phosphate concentrations through the water column. And the take home here is that phosphate concentrations tend to be extremely low in the surface waters where photosynthetic life lives. And then it increases abruptly and is present at relatively high concentrations throughout the depths of the ocean. And so I have assembled a very simple cartoon to explain why this relationship emerges. And so as photosynthetic life is photosynthesizing within the sunlit surface layers of the ocean, it's taking up nutrients like phosphate into its cells. And then eventually, these cells settle under the influence of gravity to the deep, dark depths of the ocean. And they decompose as they're being respired and providing an energy source to other types of life in the deep ocean. And as they do so, they release those nutrients back out into the environment. And so the result is that the surface layers where life lives have very low nutrient concentrations, and the dark depths tend to have high nutrient concentrations. And so keeping this whole thing going requires a mechanism to get those nutrients out of the deep ocean and put them back in the surface ocean where life can utilize them again. And that's actually non-trivial because on global average, the ocean is actually stably stratified with respect to density because we have warm, sunlit waters sitting on top of dark, cold, dense water. And so this upward motion and nutrient recycling really only occurs in special environments. So this phenomenon is referred to as upwelling, and it occurs in regions where the winds drive divergence in the surface flow. So for example, this most notably occurs off the coasts of continents where the winds are driving offshore flow. And then because the continent is there obstructing lateral flow, the requirement is that water rises up from below to replace that displaced surface water. And with it comes nutrients. And so if we look at a map of chlorophyll distributions in the surface ocean, chlorophyll being the primary photosynthetic pigment that life uses on Earth, we see that it is highly concentrated in regions of coastal upwelling and along the equator as well, where a similar phenomenon plays out due to opposing directions of Coriolis deflection on either side of the equator. And so the take-home message here is that ocean upwelling is it's a primary influence on the distribution and activity of life at Earth's surface. And I think that this is going to be true on other planets that host detectable life as well. The reason is that this relationship to upwelling does not arise from some detailed biochemistry that might be unique to Earth. This dependence on upwelling is really the consequence of the influence of gravity and the tendency for the ingredients for life to accumulate at depth in the ocean. And so there's this need for their resupply to the surface. So I should note that when I talk about detectable life, what I'm really talking about is specifically photosynthetic life. So we do in fact have chemosynthetic life here on Earth. That is life that it exists independent of stellar energy. And that life might actually be common in the universe, and in fact common potentially within our own solar system, for example, on places like icy moons. But this is a type of life that is inherently difficult to detect. And that's because chemosynthetic communities are localized, they are physically isolated from the atmosphere, and thus have limited opportunity to affect the remotely observable characteristics of a planet. And most importantly, chemosynthetic life is making a living doing the same sorts of things as geology does. It's just making a living because it does it quicker. And so these metabolisms don't tend to produce products that are uniquely biological in origin. Photosynthesis, on the other hand, has the potential to be global in scale, of course, with the notable exception of tidally locked worlds. Photosynthesis also is occurring at the surface, and photosynthetic life is in direct communication with its atmosphere, so it has the potential to influence its host planet in remotely detectable ways. And most critically, photosynthesis is ultimately taking stellar energy and converting it into chemical energy in the form of chemical disequilibrium. And this doesn't need to equal oxygen specifically, but what it does mean is that this is potentially a signal that we might recognize as weird or difficult to explain based on abiotic processes. And so, in my view, detectable life is, for the time being, photosynthetic life. And photosynthetic life is critically dependent on ocean upwelling. And so it is my hypothesis that ocean upwelling is a very important ingredient for the detectability of life on other habitable worlds. And so the question that I posed was, how will ocean upwelling differ among potentially habitable planets? And by extension, how might the detectability of life differ among those worlds? Are there certain worlds that are more likely to host very active, particularly detectable life? And are there, on the other hand, planets that are likely to host low activity biospheres that might be more vulnerable to things like biosignature false negatives? This is a sort of information that could be provide really important context for prioritizing detailed observations of different planets, or for interpreting non-detections of biosignatures. You know, it's useful to know, is this a planet that we think would be a really great host for life? Or is this a planet that might just be an okay host for life? And so what I did is I turned to a GCM called Rocky 3D. And I simulated a basically Earth-like planet. And then I systematically varied several planetary parameters in isolation. So things like surface pressure and rotation rate, orbital obliquity, and ocean salinity, to quantify how ocean upwelling, and by extension, nutrient recycling potential and biospheric productivity and biospheric detectability might scale with these planetary parameters. And so I explored a fairly large parameter space for each of these parameters. And now I'll just walk through some of the salient results of that study. So the first thing that I played with was surface pressure. And what I found was that global upwelling, which I'm defining as the sum of all upward motion at the base of the mixed layer. The base of the mixed layer can vary in both space and time, and so that's important to keep in mind. I also did this assuming just a fixed depth, like vertical upwelling at like 200 meters, and I got a qualitatively identical result. Anyways, so I found that upwelling increases with surface pressure. And this is actually a somewhat intuitive result, and that's because wind stress scales proportionally to atmospheric pressure. And it's also pretty strongly influenced by wind speed. And so you'll notice that although upwelling increases with surface pressure, it falls short of a one-to-one proportionality, and that's because wind speed is decreasing with increasing surface pressure. And so it's not clear how this relationship might extrapolate to much higher surface pressures. Yeah, and so higher surface pressure yields more upwelling, more nutrient recycling, and potentially greater productivity biospheres on these worlds. The next thing that I looked at was rotation rate. And so I varied rotation rate such that day length decreased from 10 days down to 12 hours. And what I found was that upwelling decreases with increasing rotation rate. And again, this was actually a fairly intuitive result. And that is because Ekman transport, the wind-driven circulation within the ocean, scales as the wind stress over the Coriolis parameter. And so physically, what this means is that the wind-driven circulation is this balance of the wind forcing and rotational effects. And as rotation gets faster and faster, it begins to influence things more and more. And so for slowly rotating planets, the winds are able to drive greater divergence within the surface ocean, which promotes greater upwelling. As the planet rotates faster and faster, however, winds are less effective at driving divergence and upwelling because these rotational effects become increasingly important. And so an implication here is that planets that rotate more slowly than Earth might experience greater nutrient recycling and higher productivity biospheres. Now you might be wondering about the extreme case of a tidally locked world. And so I worked with a really awesome student who was an undergraduate at UChicago at the time, now a PhD student at Harvard. And she did some simulations of Proxima Centauri B with a dynamic ocean and quantified how upwelling scales with continent area. So continents are a potential complication in that they not only shape ocean circulation patterns, but in the case of a tidally locked planet, where you expect continents to be concentrated at the substellar point, the size of the continent really comes at the expense of the area of ocean capable of supporting photosynthetic life. And so she was interested in figuring out whether continents were good news for ocean life and that they support coastal upwelling, or if they were bad news for life in that they, just limited the amount of ocean available. And so what she found was that in all cases, there was quite a bit of upwelling that occurred. So her data points here in this figure are normalized to the same Earth baseline that I normalized my study to. And so for a tightly locked aquaplanet, what you can see is that there is upwelling occurring that is 20 times greater than for our Earth-like baseline. And so that's huge. What that means is that tightly locked planets could have really strong nutrient recycling and potentially productive dayside biospheres for that reason. There are, of course, other challenges associated with tightly locked planets. And so Andrea also looked at how the mixed layer depth varies on tightly locked planets. And so she found that the mixed layer depth was generally pretty shallow for this Proxima Centauri b analog. And that's actually quite good news for photosynthesis. So here's just a quick cartoon to illustrate why. So the mixed layer depth is important because it affects the average illumination of the mixed layer. So if the mixed layer is shallow compared to light penetration depths, then that mixed layer is well illuminated. It's a great place to be a photosynthesizing organism. But if the mixed layer is very deep compared to the depth of light penetration, then that effectively reduces the amount of light that a photosynthetic cell sees through its lifetime as it's being passively mixed around within the mixed layer. It's being mixed in and out of this zone of illumination. And this is something that's particularly important on M-star planets, because on M-star planets, we expect that light penetration depths within the ocean are going to be much shallower, just simply due to the differences in the stellar spectrum. And so having a shallow mixed layer depth on in M-star oceans is particularly important for supporting photosynthetic life. And so the take home here is that Proxima Centauri b has a shallow mixed layer, which is good. That means that it's a well illuminated mixed layer, and it's got good nutrient recycling potential. And so if planets like Proxima Centauri b actually retain water and have oceans to begin with, those oceans might be good places to live. So the next thing that I looked at was how upwelling responds to orbital obliquity. And so I'm showing those results here. And you can see that upwelling does modestly increase with orbital obliquity, but the sensitivity is really quite small compared to the effects we saw from pressure and rotation rate. And so this is perhaps not a big effect on nutrient recycling through upwelling, but high obliquity planets are intriguing from a nutrient recycling potential regardless, because the depth of the mixed layer is actually varying quite dramatically on high obliquity planets. And the seasonal variability in the mixed layer depth actually provides a mechanism by which you can actually stir in nutrients from the deep ocean and put them back into the surface layers, and in a way that's not captured in our upwelling metric, because again, that's vertical motion summed at the base of the mixed layer, which can vary in space and time. And so this is a totally independent nutrient recycling mechanism. So here I'm showing Northern Hemisphere summer versus winter views of a 45-degree obliquity planet. And locally, the mixed layer depth is varying by a few orders of magnitude. in the summer, it can be as shallow as a couple of meters, and in the winter, it may extend all the way down to the bottom. And so that is a great way to get nutrients back. And so I worked with another student at U Chicago to explore how biospheric productivity actually might respond to obliquity. And so what Megan did is she took the wind fields from my GCM experiments, and she used them to force a biogeochemical model called Genie. And so she effectively just, put Earth-like life in the oceans that I was simulating. And then she quantified how that actually influences global sea-to-air fluxes of oxygen. And she did so for several different nutrient scenarios. So in this figure, I'm showing oxygen fluxes on the y-axis against obliquity scenarios on the x-axis. And each of these colored lines corresponds to a different global phosphate inventory. And so you can see, of course, that if you just increase the amount of phosphate within the ocean, you can quite dramatically increase the flux of oxygen to the atmosphere. But you can also increase the flux of oxygen to the atmosphere by changing obliquity. And in the case of if you look at modern phosphate at 0 degrees obliquity, increasing obliquity from 0 to 45 degrees obliquity actually has an effect that's on the order similar to just if you doubled phosphate in the ocean. And so this is, under some circumstances, a potentially significant effect. And I really like this study because I think it really effectively illustrates that it's not just about how much nutrient is there and available to the biosphere. It's how that nutrient is mixed around that also matters. And so I'll be doing similar experiments for all of the geophysical scenarios that I explored. We've just started with obliquity because it's the one that requires the least amount of model development to execute. I also have a soft spot for high obliquity planets because high obliquity planets introduce the possibility that we might leverage seasonality as a potential biosignature. So the idea here is quite simple. The appearance of our planet varies quite dramatically across its seasonal cycle. So the most notable feature here is the waxing and waning of snow and ice cover. And that's something that actually it's a direct reflection of the habitability of our planet because it's just evidence that we have an active hydrologic cycle. And so we might leverage a similar signal to infer habitability elsewhere. And then the other notable feature here is you see these seasonal shifts in vegetation. And these shifts actually have a big impact on atmospheric composition. So here I'm showing several years of CO2 measurements at Mauna Loa Observatory. And so of course the long-term average increase in atmospheric CO2 is the consequence of fossil fuel burning. But then superimposed on that is this really systematic seasonal cycle where we see CO2 rising and falling in response to seasonal shifts in the balance of photosynthesis, which is drawing down CO2 and incorporating it into biomass, and respiration, which is releasing that CO2 back into the atmosphere. And so throughout the warm, sunny summer months, CO2 is falling as it's being incorporated into biomass. And then that CO2 is released back into the atmosphere throughout the dark winter months. And so we might use similar seasonality to infer the presence of a photosynthetic biosphere elsewhere. And actually, I think this is potentially really powerful because if we assume that life elsewhere is carbon-based, and if we think that CO2-based feedbacks are essential to regulating climate on geologic timescales and are central to our definition of habitability to begin with, I think it's reasonable to expect that seasonality in biological activity, in biological carbon exchange with its environment, may actually manifest as CO2 seasonality on all habitable or all inhabited planets, kind of regardless of the details of biochemistry on those worlds. And so in my view, the question is not whether similar CO2 seasonality is happening elsewhere. The question is really whether or not that seasonality might be detectable. And so there are, of course, some big challenges there. Among them is that CO2 is extremely soluble because it forms a number of species in seawater. And so CO2 seasonality arising from the marine biosphere is actually chemically buffered and ineffectively communicated to the atmosphere. So if we look at CO2 seasonality from the northern hemisphere versus the southern hemisphere, which has considerably less land area and correspondingly more ocean area, we see that the northern hemisphere signal is several times larger than that of the southern hemisphere signal. And that's due to this buffering effect within seawater. And so although the focus of this talk has been on the role of ocean dynamics and the importance of oceans for life, I think it's important potentially that we have continents, because in this case the detectability of CO2 seasonality is likely to be strongly dependent on whether or not there's land on a particular planet. Continents also, have additional importance. So again, continents drive coastal upwelling and nutrient recycling within oceans. I haven't explicitly pointed this out yet, but on each of the figures that I have shown you, you might have noticed that the coastal upwelling fraction represents the largest upwelling fraction for every scenario. And so if you didn't have continents, nutrient recycling potential would be severely muted for that reason. That's problematic because in the absence of nutrient upwelling, the major source of nutrient supply to the mixed layer is really from just from weathering, and that's a relatively small flux. There are some seafloor processes that can provide nutrients to the ocean, but they're relatively insignificant on Earth today. And so if we got rid of the continents, you just have the seafloor flux. You might imagine scenarios where that flux might be greater, but there are also scenarios where that flux might be even smaller than it is on Earth. So for example, if you don't have continents because you have a lot of water, 100 oceans worth of water, then you run into this issue where high density ices become stable and might act to actually physically isolate water from rock at the base of the ocean. And so that would shut down that nutrient flux altogether. And so it is my view that water worlds are potentially really bad places to live, even though they have lots of water and may technically meet our existing definitions of habitability in that regard. I think that there are additional biological challenges to living on these worlds. Okay, so The last planetary parameter that I'll talk about is ocean salinity. So I varied ocean salinity from 10% to double modern Earth's salinity. And this did have a small impact on ocean upwelling. But like obliquity, it's a relatively small effect. And that's particularly true compared to other impacts that it had on the planet. So, for example, salinity had a huge impact on planetary climate. So doubling salinity with respect to present-day Earth actually completely eliminated sea ice and resulted in 6K warming on global average. So that's huge. And so I was actually quite surprised by that result. But perhaps I shouldn't have been, because there are actually several good reasons to expect that salinity should influence global climate. So for example, salt directly impacts the density of seawater. You add more salt, seawater gets denser. And as that's happening, it's also changing the relationship between temperature and density. So fresh water is most dense at 4 degrees Celsius, whereas very salty water just keeps getting denser and denser as it gets colder. And so in combination, what that means is that salinity can really affect the density structure of the ocean and its circulation and ocean heat transport. And so that's potentially an important climate consideration. Meanwhile, salt also suppresses the freezing point of seawater, and so that's something that can directly limit sea ice formation. And then trace inclusion of salt in the resulting ice that may eventually form affects seasonal melt pond dynamics and the average albedo of sea ice as a result. And then of course salt, these effects are not included in rocky 3D, but salt also affects evaporation and can serve as a sea salt aerosol. Can be CCN. And so these are all things that can influence climate. And so I got excited about this with respect to Earth's climate system and thinking about the role of salinity in our own planet's climate through time. In particular, one of the questions that fascinates me is the so-called Fate-Young Sun paradox. So for those of you who aren't familiar, this orange line is showing the trajectory of solar luminosity through time advancing to the right. So our sun has gotten increasingly brighter through time. Now in the blue shaded region, what I'm showing is the lower and upper estimates for Earth's surface temperature corresponding to, just if we had no greenhouse effect, providing the lowermost limit, and if we had current greenhouse effect, providing the upper limit. And the point here is that unless we had a considerably greater greenhouse effect, our planet should have been well below freezing for more than half of its history. And we know that that wasn't true. We have compelling evidence for liquid water going basically all the way back. And so most explanations for explaining this, have simply focused on atmospheric chemistry and just said, well, CO2 was much higher. And that's, almost certainly true that CO2 was much higher. But I was interested in seeing, if salinity was higher, is that something that could have also compensated for reduced solar luminosity? And so I dug into this, and I was disappointed actually to find that Archean salinity is really poorly constrained. all of the kind of theoretical estimates suggest that Archean salinity should have been higher than today. So, for example, Dick Holland noted that if you just take all of the known evaporite deposits on the continents today and you just dump them back into the ocean, well, then ocean salinity should have been much higher than today, perhaps 1.5 to 2 times present day salinity. And of course, from my exoplanet simulations, we know that doubling salinity can have a huge climate effect. Unfortunately, the geochemical proxies are extremely sparse and fairly controversial. And they basically permit salinity that was somewhat lower than or somewhat higher than today. And so I was interested in characterizing the climate uncertainty that arises from this salinity uncertainty. And so I simulated Archean climate for 3 different salinity scenarios, a low salinity scenario, a 20 PSU salinity scenario, and a high salinity scenario, 50 PSU, and then of course a modern salinity scenario. And what I found was actually pretty shocking. So here I'm showing different salinity and CO2 scenarios, salinity in the columns and CO2 scenarios in the rows. And so the first thing to note is that as you increase CO2, your climate does indeed get warmer. So moving from the bottom row up to the top row, there is consistently less sea ice. But salinity is also having a big effect. So as you move from left to right, you also get consistently lower sea ice cover under higher salinity conditions. And if you have both high CO2 and high salinity, you can actually simulate a planet that is warmer than at present day, despite receiving less energy from the sun. So I have a paper in review at the moment arguing that ocean salinity might be an important consideration for early Earth climate, in addition to exoplanet climate. But I think it's also really interesting to note that it's not just salinity that has changed through Earth's history. Many of these other things that I've been talking about today have also changed through Earth's history. So for example, surface pressure has changed potentially quite dramatically. Estimates range from less than half a bar in the Archean to 2 bars in the Archean. So that's potentially has important biological implications. And rotation rate has also changed through Earth's history as well. And so just briefly, in my last few minutes here, this curve is showing Earth's oxygenation trajectory. And then this panel here is showing the evolution of day length on Earth. Now, we know that the origin of oxygenic photosynthesis really quite dramatically predates the achievement of high levels of oxygen in our atmosphere. By some estimates, it may have been 2.5 billion years between the origin of oxygenic photosynthesis and the achievement of really high oxygen levels in our atmosphere. And we don't really know why. Most people are leaning towards low nutrient inventories throughout the Proterozoic, the so-called boring billion. But there's not really mechanistic reason to think that phosphate levels should have been dramatically lower. But what I would like to propose is that perhaps phosphate recycling potential was lower because early Earth was rotating faster. And so to illustrate that, we know that rotation rate has slowed through time, and we think that atmospheric pressure has potentially increased through time. We know that continental area has also increased through time, and so perhaps the sum of these differences has been to promote greater upwelling, greater nutrient recycling, and greater biological oxygen production, and thus, a protracted oxygenation of Earth's atmosphere. And so I recently got funding from the Habitable Worlds program to explore these relationships through Earth history. And ultimately, doing so is a great test of the hypotheses that I've laid out in the exoplanet realm, to see if certain worlds are just more likely to become oxygenated by their biospheres than others. So to summarize real quickly, I have shown you that ocean circulation is a key constraint on the distribution of photosynthetic activity at Earth's surface. And I've shown you that nutrient recycling potential will differ among potentially habitable exoplanets and on early Earth as well. And so for that reason, some oceans may be better places to live than others, or at least more likely to host detectable life than others. And in particular, those planets that might be particularly hospitable towards photosynthetic life, thus detectable life, might be those that have higher surface pressure, slower rotation, more extensive coastline, particularly north-south oriented coastline, higher orbital obliquity, and higher ocean salinity. And so it's my view that not only do all of these things increase the likelihood that a planet might be a good place to live, all of these things may increase the probability that life on that planet might actually be remotely detectable. And so with that, I will thank you and accept any questions.
Thank you so much, Stephanie. That was great. That was a really great talk. Yeah, so if anyone has any questions, feel free to raise your hand. And it looks like Prabal, you have a question. So if you can unmute yourself and just introduce yourself.
Sure. Hey, Stephanie. It was a great talk. My name is Prabal Saxena. And actually, one of the things I was wondering about was kind of the frequency of upwelling. So I know if you think about like thermohaline circulation, you have different frequencies for a lot of this type of stuff. And when you look at other planets and they have these maybe shorter rotation periods if they're tidally locked, how does that end up affecting kind of nutrient delivery and things like that?
Ooh, that's a great question. So the specific example of tidally locked worlds, we've looked at that a little bit with the specific example of Proxima Centauri b, but that's the slower rotation, and remember, not the faster that you asked about. And so I don't have a great answer for that, but it's certainly something that could and should be explored.
Great. It looks like Rick, you had one.
Yeah, yeah, nice talk. Really interesting stuff. I was wondering, like, going back to more of Ekman transport in general, like you had a slide showing that all of the Earth, like a lot of the phosphorus, is along the coastlines, and you really talk about the importance of coastal upwelling, which is really important on the surface winds, and it varies like quadratically in regards to that. So when you were doing all of your explorations and you were changing the rotation rate, what changed in the surface winds during those explorations?
That's a great question. So the winds did not change as significantly as the ocean response to the winds. I have a figure that I can send you that looks specifically at the winds, but the response is really dictated by the rotational effects within the ocean rather than differences in the wind forcing there.
Okay, yeah, I was just sort of curious, is that did you use in Rocky 3D, are you using the prevailing winds now on Earth, or did they spin up in a
Oh,
new
yeah, yeah.
atmosphere
So
every
we calculated self-consistent winds. We had it was a fully coupled ocean atmosphere GCM.
Oh, cool. Okay.
The winds are responding to rotation rate as well. They're just not as important.
Cool. I look forward to the paper. Thanks.
And I think, Vlad, you had a couple of questions?
Yeah. Hello, Stephanie. Yeah, thanks for a nice talk. Yeah, I have a couple of questions and a comment. The first question is that how does the rate of photosynthesis depend on the temperature, the water temperature, and the air pressure? That's the first question. The second one, how does the CO2 solubility depend on the temperature?
Sure. So
Pressure.
both photosynthesis and CO2 solubility depend on temperature. Photosynthesis, ooh, it's not a linear relationship for either of those things. The CO2 solubility will also scale with well, not the equilibrium solubility, but the gas exchange also scales with wind speed quite dramatically, and so that depends on wind speed squared, and so that can be a bigger effect than temperature under some circumstances. But I think at some point, low temperature can limit biological phosphate utilization, but at high temperature, you don't get increasingly more photosynthesis just because it's hotter. You're ultimately limited by that phosphorus. So that was kind of circular, but did it answer?
Yeah, because as far as I remember, at temperatures above 20 degrees Celsius, the photosynthesis goes down. So that's what I remember. I don't, so there is a certain limit on temperature.
Yes. Life exists within a relatively narrow temperature window on Earth. If temperatures above 20 Celsius, but life typically has a preferred temperature. What is unclear is the extent to which that preferred temperature is kind of dictated by the prevailing temperature in the recent past, and whether, if Earth has had a different climate trajectory, maybe that preferred temperature wouldn't be less than 20 degrees, right? Maybe it would be higher or lower, depending on what's happened through our planet's history.
And second was about the CO2 solubility.
Yeah, so solubility absolutely responds to temperature. So of course, solubility decreases with increasing temperature. It'll also decrease with increasing salinity. So saltier oceans might be more favorable for things like CO2 seasonality arising from marine biospheres for that reason. But that relationship is not linear, and it's complicated by the fact that the winds are responding to climate as well, and that'll affect gas exchange kinetics quite dramatically.
Yeah. Thank you. And the comment that I want to make, it's regarding your point on the high salinity, which is very interesting. So, it not only has the effect on climate of the Hadean Earth or Archean Earth, but also on the rate of prebiotic chemistry reactions. It has been proven in laboratory experiments that the rate of biochemical reactions, depends on salinity and the pH. So the greater salinity, the faster the reactions, which is an important point too.
Yeah. That is something that I've been thinking about a lot, because on the one hand you think you have more ions in solution, you can do more chemistry. On the other hand, there might be an upper limit where now you've got too much And so I'd be really interested in knowing if, if there's an optimal salinity for the origin of life, and if it's something in range of what is reasonable for early Earth ocean chemistry. Unfortunately, we don't have great predictive models for what early Earth salinity should have been. There's this issue of both changing fluxes of salt in and out of the ocean, but then also over geologic timescales, the amount of water in the ocean probably has also changed as well. And so it's a tough question, but it's something that I'm really excited about these days. And actually, I'm looking to hire a student or postdoc to work on.
All right. It looks like we have time for maybe one last question. I think there might have been one from Gavin in the chat, Stephanie. I don't know if you can see that, but it looks like they were asking about ocean heat transport.
Let's see. Changes ice albedo feedback through freezing point change, but what about? Yes, changing salinity absolutely changes ocean heat transport. Ocean heat transport is enhanced at higher salinity. There is greater overturning circulation occurring.
That's great. Well, we are approaching the end of the hour, so I just want to thank Stephanie again for that great talk. And if you have any other questions, feel free to direct them. I can send out your email if you need to. I'm going to go ahead and post the schedule for the remaining seminars in the chat. So be sure to take a look at that so you can see what other ones are coming up this year. And thank you again, Stephanie.
Yeah, thanks for having me. This was fun.
Thanks.